Welcome, everybody. My name is Brian O'Hare, and I am the Terminology Maintenance Manager for the MSSO. Next slide, please. So first of all, I want to thank you for registering this event. Just a couple housekeeping things that I wanted to make you aware of. First of all, a number of you have asked prior to this meeting if this session is being recorded, and it certainly will be. Barring any technical issues with the recording, we will be posting it to the MSSO's YouTube channel roughly 24 to 48 hours after the event. There will be opportunities to ask questions during this meeting at the end of each presentation. And at the end of the meeting, we have an open session for questions. To ask a question, you need to expand the control panel located in the upper right of your screen to access the questions pane and type your questions in there, and then we will address those questions as best we can during the meeting. Next slide. So this is our agenda for today. We have a full agenda comprised of a mix of speakers from industry, academia, regulatory authorities, and from the MSSO. Next slide. So before we start with our first speaker, I wanted to inform everyone that the Uppsala Monitoring Center and the MSSO will be holding a joint WHO Drug Medra global meeting on the 22nd of June from 2 p.m. to 5 p.m. Central European Summer Time, which is 8 a.m. to 11 a.m. Eastern Daylight Time in the United States. This is a virtual meeting, and there are a number of topics that are planned, including a coding workshop and a presentation and panel discussion on the results of a medical coding survey, of which we have a brief explanation of in today's agenda. The registration for the global event will be announced in the days following today's user group meeting. So without further delay, let me introduce our first speaker on the agenda, Patrick Revel, the Director of the MSSO, who will be presenting on the topic of Medra interoperability. Pat, the floor is yours.
Great. Well, thank you, Brian, and good afternoon and good morning to all the attendees. Very much appreciate you making the effort to join. And I'd like to focus a bit just on Medra interoperability and give you a sense of what is available if you don't already know that. So next slide, please. So what is interoperability? And just make sure we're all talking about the same thing. It's defined as the ability of different computerized products or systems to readily connect and exchange information with one another. In the context of medical terminologies and what we're working with, it's the ability to convert data coded in one terminology to another. Now, I think you always have to be careful in using mappings or with interoperability that it's not perfect. So during a conversion process, it is possible that mappings can introduce a potential loss of information. So I'll talk a little bit more about that and maybe give you some examples of what I mean. So next slide, please. So these are the currently available mappings that we have related to Medra. The first mapping, the WHOART to Medra bridge, is primarily used to convert data from the older terminology called WHOART into Medra. And it's a one-way mapping from WHOART into Medra. If you remember, WHOART was originally developed by the Uppsala Monitoring Center, and it was used pretty widely, and people still do have data today that is coded in WHOART that they would like to convert. So we maintain that mapping. We originally built it with our colleagues at the Uppsala Monitoring Center, but we've since they've stopped maintaining and developing that terminology, we just maintain the Medra side of it. So that's certainly available for people to use, and we provide that to people if they'd like to use that one. The others are all active terminologies and either integrate Medra directly into their terminologies or map their terms to Medra terms. So, for instance, the CTCAE terminology uses Medra terms directly, where IMDRF has a IMDRF term, which they in turn map to Medra. I'd like to discuss one of the current mappings, the Snomed CT and Medra mapping, and then mention another few mappings that are currently under development. So next slide, please. So the Snomed CT Medra mapping, initially released last year, roughly this time last year in April of 2021. We just over a week ago provided an update to that mapping. It's based on Medra 24.1 and the January 2022 release of the Snomed CT International version. You might notice in the case of the Medra release, it's one version back. We have to identify a version where we can do the update. So in this case, it's one version back, but I don't think the mapping is significantly impacted by that. This recent update included roughly 250 new terms that were mapped, including some relevant COVID-19 terms. And as you might expect during a course of this sort of an update, the changes result from version changes in both terminologies, whether that means a new term that's been added or changes in currency in the context of Medra, or inactivations in terms of Snomed CT. So next slide, please. So switching gears now to ICD, and I'm going to talk about ICD-10 and then ICD-11. The Medra Management Committee has supported the development of a mapping from ICD-10 to Medra, and it's only in that direction, so ICD-10 to Medra. And just to be clear, what I'm talking about is the International Classification of Diseases, otherwise known as ICD-10, the 2019 international version. You may also be aware of other versions that are nation-specific versions. You know, the US has ICD-10-CM, where they essentially extend the terminology to add more detail, but they all share a common root, which is this international version. ICD terminologies are widely used for mortality and morbidity statistics, as well as a great deal of registries use ICD-10. So we think it will be a great source of data that you can pull from these other sources and convert into Medra. So we're working now with WHO and ICH to develop an agreement for the distribution and maintenance of this mapping. And we look and we're hopeful that fairly soon, and hopefully before the end of the year, we could have this mapping available to users. Next slide, please. So a little bit more detail about the ICD-10 mapping. It is roughly 11,000 terms. But we've also added, I think, a very useful feature, which is the idea of an attribute that describes the individual mappings. In other words, when you look at two terms that have been mapped, what is the relationship? Are they, in this case, are they equivalent terms? In other words, there's no loss of information when you're using that mapping. Or is there loss of information? So you can see for specific maps whether there's a potential loss of information. The smallest subset is really a few terms that were unmapped because of the scope of the terminology. So that at times, because ICD-10 is a bit different than Medra, or Medra is a bit different than ICD-10, there will be some terms that are unmapped, but the vast majority are mapped in some form. So talk a little bit more about the attributes. So you can see the column headings that are in green show the ICD-10 terms, the column headings in blue show the Medra terms, and then you see the attribute all the way on the right-hand side. So just to give a few examples, you can see the first one, yellow fever unspecified, is exactly the same between ICD-10 and Medra. So that one's a very simple one to say those are equivalent. Now, in the second case, birth injury to facial nerve, is very similar to facial nerve injury due to birth trauma. So those are seen still as equivalent, even though their text is slightly different from each other. The third entry, measles with other complications, is mapped just to measles on the Medra side. So you could see in that case, we're missing the concept of other complications. So that one is marked as loss of information. And finally, the last one that's listed there is a pedestrian injured in a collision with a Fetal cycle. That doesn't exist in Medra, so that one is essentially unmapped and certainly not available. So this spreadsheet format or something very similar to it will be available to Medra users from the Medra.org website, and then we will work to maintain this mapping with our colleagues at WHO. So Icd-10 is really no longer growing or changing, although there are some infrequent changes to it, but certainly the Medra side of that will change. So it's important, I think, to maintain that mapping going forward. So next slide, yep, thank you. So let me shift now to Icd-11. So the Medra Management Committee has also supported the exploration with WHO of a development and maintenance of mappings between Icd-11 and Medra. Now, unlike the Icd-10 mapping, this one will be bidirectional. In other words, you would be able to convert from Icd-10 to Medra and also from Medra to, Icd-11 to Medra and from Medra to Icd-11. It's a bit of a larger task because Icd-11, if you're familiar with it at all, is a bit larger. So it'll take a little bit more time, but I think it's, again, will be a useful product to have in hand. Certainly is important, I think, because both terminologies will be changing, that these will be updated on a regular basis as the terminologies change, and quite frankly, as users might have some input about what we have produced for the mapping. So you as a user will have an ability to comment or suggest changes to existing mappings that have been developed. So that's certainly a part of what we see as the maintenance process in coordination with WHO. So more details to follow about the Icd-11 mapping, but I think we're very excited to be able to produce both of those, hopefully in the near future. So next slide, please. This slide, I don't expect you to be able to see all the detailed text there, but I just wanted to point out we do have a page on the Medra.org website devoted to mappings. And the text in the middle, which is the unreadable part, is, it gives a general description of the mappings that are available. And on the right-hand side, you'll see various links to where those mappings are located. In most cases, they're not on the Medra website. They're in other sources where the owners of those terminologies house those mappings. So I really encourage you to take a look at these and see which ones of these mappings would be helpful to you. So next slide, please. And this is essentially just a mapping reference with some of the same information. So you can see all of the mappings that I've listed earlier in the presentation in the leftmost column, a description of the mapping, the maintenance process, in other words, who is in charge of maintaining it, where the MSSO works with these organizations to maintain these terminologies. And finally, the links where you can actually access these mappings. So again, I see this as a good reference, and if you can find a utility for it, at least you can be aware of what is available from a mapping perspective. And I think with that, I'm open to any questions or comments you might have.
Okay. So if anyone has any questions, they're free to put them into the questions pane. We'll wait a few seconds here to see if we have any questions. None at this time.
Okay. Well, I'll be available at the end if there are other questions as well.
Okay. So not seeing any questions at this time, so why don't we go ahead and move on to our next presenter. who is Professor Anna David, Director of the University College London Institute for Women's Health, and she will be delivering a presentation on maternal and fetal adverse event terminology, a new system to provide better safety monitoring for clinical trials in pregnancy. So Professor David, the floor is yours. I think you're on mute. We cannot hear you, Professor David. I think you're muted.
Can you hear me now? Apologies, my computer just hung at the critical moment.
Yes, we can. Thank you.
Brilliant. Okay. Can you see my screen?
Yes, we can.
Okay, perfect. It's always typical that these things happen just as you're about to start speaking. So hopefully that won't happen again. So thank you very much for the opportunity to speak at this Medra users group. My name is Anna David. I'm an obstetrician and fetal medicine specialist, and I work at University College London in the UK. And what I'm going to speak to you today is about MFET, or maternal fetal adverse event terminology, which we've been developing for the last 7 years. My disclosures are here. I'm co-chair of the Maternal Health Project of ABPI, and I also do some consulting for MedDRA as a private non-for-profit developing a prenatal therapy. So as an obstetrician fetal medicine specialist, when I'm thinking about treating conditions in pregnancy, I think about the general health of the woman, the mother, but also her fetus and also her future baby, her neonate. And we have some particular general health problems that she might have. She might develop some general adverse events, or she might have some pregnancy-specific conditions. For example, preeclampsia with high blood pressure and protein in the urine, preterm birth going into labour early, fetal growth problems, either large baby or a small baby, placenta problems, problems to do with the liquor or the amniotic fluid around the baby, and obviously problems that can occur at the time of delivery. Now, my work over the last few years has been trying to develop a treatment for a condition called early onset fetal growth restriction, manifested here by this little girl, Abigail, who was born at 27 weeks of pregnancy, weighing 1.5 pounds, and was really born at around half the weight that she should have been. This is her father's hand here. And I was part of a group leading a team funded by the European Commission to try to develop a new treatment for this condition. And this treatment was to give a gene therapy to the mother to increase blood flow to the uterus to overcome this condition, to make the baby grow better. And during this 6-year program, we had a bioethics study to look at the ethical conditions and whether it was possible to actually give mothers gene therapy. We did a reproductive toxicology. We manufactured and tested a new drug product, and we developed a first-in-woman phase 1/2A safety efficacy study. And it was during this development of the clinical trial protocol that we realized it was going to be very, very difficult for us to measure safety in this particular trial. And that was because there was very little in terms of safety signals for us to go on. And if we're developing a new first-in-human drug, we need to really be certain, primarily our primary outcome measure was going to be safety. So we had a look at what was available in the terminology at the time, and we had a look at CTCAE. In fact, the version we looked at before at the time was version 4, but we now have version 5, which was November 2017. And there are lots of adverse event definitions that you can look at, but at the time there were only 4 definitions for pregnancy. So fetal death, premature delivery, fetal growth retardation, or pregnancy, puerperium and postnatal conditions other. In other words, there was very little that we could use to assess safety in the mother and particularly assess safety in the fetus. And we were going to want to measure safety in the fetus to find out whether this new treatment was going to be beneficial or harmful. We also had a look at the Division of AIDS, version 2.1, July 2017, that lists adverse events with severity grading. And you can see here there were 3. Stillbirth, preterm birth, or spontaneous abortion or miscarriage. So again, we have a problem trying to get a handle on adverse event safety in the mother and in the fetus. We also had a look at what categorization the World Health Organization had, and it classified severity of preterm birth according to the gestational age of delivery. So, for example, extremely preterm, less than 28 weeks of gestation, very preterm birth at 28 to 32 weeks, and then moderate to late preterm 32 to 37 weeks. We also looked widely and found the Clavian Dindo classification of surgical complications. The particular drug that we were going to be using was going to be given into the maternal uterine arteries using interventional radiology. So this was relevant to us. And there was grading, as you can see here, grade 1 to 5. And that was useful from a sort of interventional point of view, but again, didn't give us any information about the fetus. So the variety of issues with adverse events in pregnancy. Events can have a very different impact on the pregnant woman and the fetus. And just to give you a couple of examples, the woman can potentially develop an infection, perhaps an infection in the fluid around the baby, so chorioamnionitis, which might actually be very mild, but could end up causing her to go into labour, could end up infecting the fetus, and the fetus actually to have a grade 5 to die because of this adverse event. On the other hand, the mother may have a very severe problem. She might have a major hemorrhage, and that might then cause the fetus to be delivered a little bit early, but not to have such a major impact. Again, adverse event grading is often based on the need for hospital admission. We have a very low threshold as an obstetrician for admitting pregnant women for observation. For example, raised blood pressure. We might bring somebody into hospital so we can monitor their blood pressure regularly over 24 hours, and then we might discharge them again without actually giving any particular definitive treatment. And obviously many women will already be in hospital when they give birth. So grading adverse events just on a hospital admission is probably not going to be that helpful. But the most important thing is it's very difficult to assess the impact of any intervention on the fetus. You've got to be able to monitor what actually the fetus is doing. How are they? And, you know, it's a black box in there. We have very little information that we can gain. But there are some things that we can do. For example, we can assess the fetal heart rate. We can assess how much the uterus is contracting through a cardiotocography or a CTG, often called a non-stress test. We can image the fetus and the placenta, we can do ultrasound, we can do MRI, we can occasionally do CT if we need to. And we can also monitor fetal movements. For example, we can ask the mother about fetal movements, we can put a pressure gauge on her uterus and measure fetal movements. So it's not completely impossible to do it, it's just not very easy. And so we developed this system, which we call MFET, Maternal Fetal Adverse Event Terminology, which we published in Prenatal Diagnosis Journal in 2021. This is the link to the paper. We have a whole load of resources available on our website at UCL. And I'm most grateful for Rebecca Spencer, who's a PhD student who worked with me, who helped me develop this terminology, as well as the steering committee who worked really hard on this for about 7 years. I'm going to tell you about how we developed this terminology and then give you some examples of how you might use it. So back in 2015, we reviewed all the existing grading criteria and also looked at relevant national and international guidelines to come up with some definitions of adverse events. So for example, we looked at guidelines from the American College of Obstetricians and Gynecologists, the Royal College, the Australian New Zealand College, International Society of Prenatal Diagnosis, SMFM, BMFMS, so these are fetal medicine societies, ISUOG, World Health Organization. Then, for example, you can see gestational hypertension, preeclampsia and eclampsia guidelines. These are all the guidelines that we were able to identify, come up with, a variety of them from WHO, ACOG, ISSHP, Canadian guidelines. We have Mexican College. We also very kindly were able to access some clinical trial protocol from GlaxoSmithKline for a drug called Retrieval. And so we had a variety of definitions that were available for maternal adverse events. We then met as a consensus group, and this was a combination of obstetricians, obstetric trialists, fetal medicine experts, fetal surgeons, pediatric surgeons, neonatologists, and industry representatives from GSK. And we basically sat down over 2 days and went through and decided how we're going to assess adverse events, mainly in the fetus. So we came up with a system that focused on organs, and the particular organ of the fetus. We looked at the heart, we looked at the brain, the kidneys, movements, and we defined them as you'll see later. We came up with a draft set of 12 maternal and 18 fetal adverse event definitions and the severity criteria. We then liaised with Medra MSSO very kindly and liaised and integrated them into the Medra terminology. We aligned them to make sure that they were correct and added on to Medra version 19, published in March 2016, these new fetal terms. We then also wanted to make sure that these adverse events were really important for patients. Were we missing anything out that patients felt were useful and that we as clinicians potentially were not thinking about? And so we convened a patient public engagement group that I already worked with as part of a project called Gift Surge. And these were charity representatives broadly from across various different groups in the UK. And they came up with some good practice recommendations, not some new adverse events, but some definite things, food for thought when we're defining and producing clinical trial protocols. They felt it was very important that any antenatal decisions by the woman or the couple to terminate the pregnancy or to have only palliative neonatal care after birth was going to be very important to document. So, for example, a condition might be affected by a drug or an intervention. The patient might decide to terminate the pregnancy because of that, or not to have any intervention after the baby was born. And very important to record those decisions so that you don't bias your data. They felt it was important to report the mode of onset of labour and the mode of delivery, including whether the mode of delivery that the patient underwent is likely to impact future pregnancies. So, for example, an intervention, a drug, might be more likely to cause the woman to need a caesarean section, and therefore that might affect how she was delivered in future pregnancies. Another area that they felt was important was to assess the psychological impact of the intervention on the pregnant woman, including consideration of the psychological impact of any fetal adverse events. So how does what happened to the fetus potentially affect her stress and anxiety? And that they felt we should be using validated measures in comparison with an untreated group to monitor these psychological impacts. Where possible, to include an assessment of the fetal response to an intervention, including indications of fetal pain or stress. That's very difficult to measure. We don't actually have an adverse event definition or grading for this, but I think it's something that we really do need to consider and work on. And they felt it was important to record data on subsequent fertility and pregnancies over a time period proportion relevant to the intervention. So, for example, if a drug caused a woman to deliver early or to have complications during her pregnancy, which meant that she was less likely to be able to conceive in a future pregnancy, then that was going to be important information to collect. So I don't think many of these actually affected our terminology, but it's very worthwhile for anybody who's developing clinical trial protocols out there, which it'd be useful to collect up this kind of information. So we then subjected our terminology, our grading, to two-phase Delphi. We had separate online surveys for maternal and fetal adverse events, and we had in our fetal adverse events 63 participants in our first round, 54 in our second round, to clinicians, scientists, industry, midwifery, patient, charity representatives, and you can see here it was international. And again, for our maternal adverse events, we had 45 participants in our first round, 39 in our second round, and again, the same types of people taking part, and again, it was international. And when we'd finished that, we had more than 70% agreement achieved for all of our 31 definitions, 97% agreement for our maternal severity grading, and 92% for our fetal severity criteria. We then had a final meeting of the steering committee and a remote discussion to address the 8 outstanding issues and issued and developed this terminology version 1.0. We've since in February aligned it with Medra again, and it's now version 1.1 with some slight tweaks to the phrasing in the actual terminology. So some general principles in terms of grading. We are grading the severity independently for the pregnant woman and the fetus in some cases for adverse events, as I'll show you shortly. As I said, pregnancy conditions can affect the mother and the fetus separately, and that's the rationale for this. But the generic fetal adverse events are based on the CTCAE generic criteria, which goes from grade 1 through to grade 5. You'll see here grade 3 is hospitalization. But when you're a fetus, you can't be separately hospitalized from your mother. And it could be that you might have a very severe fetal adverse event, but that doesn't compromise the maternal condition. And so therefore using a hospital admission for a fetal adverse event is probably not very helpful. And so we don't have hospital admission in our grading system. We do have in our severe grading increased frequency of monitoring, which might require women to be admitted to hospital, but not necessarily. But it would be likely to lead to significant neonatal morbidity. Grade 4 is life-threatening, likely to lead to fetal injury or permanent disability, or to lead to neonatal death, or a change in management, and then fetal death is grade 5. Now, of course, adverse events in the fetus need to be defined as diagnosable in utero. So they're not something that you would be unable to pick up in the fetus. They might continue in the neonate, but you have to be able to diagnose them in the fetus with potential to cause detriment to that fetus. This is the list of the adverse events. We have 4 at the top that are similar for the mother and the fetus. So hemorrhage in pregnancy has an effect on the mother, but also has an effect on the fetus. Preterm premature rupture of the membrane, similarly, chorioamnionitis and anemia of pregnancy. And then you can see our list of maternal adverse events here, hypertension, premature labour infection, hemorrhage, retained placenta, and amniotic fluid embolism. And then on the right-hand side, we have a variety of different conditions in the fetus: fetal fluid collection, conditions of abnormal fetal heart rate, bradycardia, not labour, so this is non-labour, this is antenatal bradycardia, tachyarrhythmia, cardiac function abnormalities, then some abnormalities in imaging, so of the brain, the gut, the muscle, the kidneys, fetal movement abnormalities, fetal neoplasm, so a tumour, fetal structural abnormalities not otherwise classified, abnormal fetal growth, and then procedural hemorrhage and post-procedural hemorrhage. And just to give you some examples here, this is gestational hypertension, the definition: new onset hypertension, presenting after 20 weeks gestation in the absence of indicators of preeclampsia. We had 100% agreement, and in terms of our grading, we had 97% agreement for our grade 1 and grade 2. This is our definition of preeclampsia: new onset hypertension presenting after 20 weeks with 1 or more indicator of preeclampsia. We had 95% agreement for the definition, and in terms of the grading, you can see here the range for all of them over 70%. Again, grade 5 is maternal or fetal death. Then we had eclampsia as well, generalized maternal seizures between 20 weeks gestation up to 28 weeks after delivery, not attributable to any other condition. Agreement of 84% for this definition, and 89% for eclampsia being graded as a grade 4 life-threatening adverse event. And you can see here maternal anemia of pregnancy and fetal anemia of pregnancy. So in terms of this disorder characterized by a reduction in the amount of hemoglobin in the blood occurring during pregnancy or the perioperium in the absence of hemoglobinopathies. And sorry, just to go back there, it affects the mother and the fetus in different ways. So in the mother, there's a wide range of grading, but in the fetus, you would only pick it up, we would only define it as a grade 4 life-threatening if there was pathological signs on the cardiotocography or fetal indication for delivery. In terms of specific fetal adverse events, some examples here: fetal fluid collection goes right the way through from grade 2 up to grade 5, fetal death, life-threatening grade 4, new onset accumulation of fluid in at least 2 fetal compartments, i.e. what we call fetal hydrops, or life-threatening isolated pericardial, pleural, or peritoneal fluid collection. And then fetal cardiac abnormalities defined as an abnormality in fetal cardiac function, and grade 4 here, likely to lead to fetal injury or permanent disability, requiring a substantial change in management, including potentially delivering. So this is the web page here where you can get hold of the resources that we've put up there. We have a PDF which explains all the definitions and all the gradings, and we've also been liaising with Medra, and they very kindly help us produce an Excel spreadsheet. This is an example of all the maternal adverse events. You can see the adverse events, the lower limit terms here, the code, and then the grading and the definitions. And this is for the maternal adverse events. And again, for the fetal adverse events, you can see the code and the definitions as well. So in terms of the future, we realise that this is really a work in progress. This is a start. We're very keen to disseminate and promote the system. We're working with regulatory authorities. We've been in touch with FDA, EMA, MHRA. We've aligned version 1.1 with current Medra terms to make sure that it's really mapped properly. And our vision is to include it in all protocols of trials in pregnancy, because that's the way that we're going to get the ability to measure safety in women and their babies. And of course, it's not just for clinical trials. If you're doing any kind of observational studies, you can use the terminology to grade your maternal fetal adverse events in those studies as well, and to compare drug and surgical interventions. Again, this is the link to the resources page. We have a steering committee set up, and we will be shortly posting on there a system so you can give us feedback. We'll be reviewing the terminology every 6 months to decide what we need to add. We realise that we're going to need to add terminology. People may come up and say, oh, we're not sure about that grading. But if you have an adverse event that you feel is not actually captured by the current terminology, you can use the generic fetal adverse event grading system, which is also available. So in conclusion, MFET is a comprehensive standardized system to define and grade maternal fetal adverse events. It's aligned with Medra in version 1.1. We've used an international Delphi process over a number of stages to define the grading. We think it's going to allow systematic and consistent grading in clinical trials in pregnancy, and hopefully this will provide meaningful understanding of safety and to allow trials to be compared. And I'm very grateful to all our steering committee, especially Rebecca Spencer, our patient engagement groups, charities across the UK, our funding, including NIHR, European Commission, and Wellcome Trust and EPSRC. Thank you very much for your attention, and I hope there's a bit of time for some questions.
Yes, we do have a couple questions. The first question is, did you have any maternal or fetal adverse events that were directly related to COVID-19 or any studies with this?
So we haven't, no, we haven't developed any specifically, because I think they would be picked up by the sort of generic terms. I think that, because you can't, the only way that you'd be able to pick up whether the fetus had COVID-19 would be to put a needle into the fetal blood and collect a sample. As you have to be able to measure what is going on in the fetus. Of course, if COVID-19 caused, let's say, for example, the fetus to become anemic, and then you found that they had high drops, then of course you'd be able to identify that there was an adverse event from that point of view. Does that make sense?
Okay, great, thank you. Okay, the next question, this is more of a specific Medra question, so we'll see if we can answer this. Question is, how do you recommend to use your maternal and fetal adverse event terminology with, or versus, I should say, versus the Medra, the standardized Medra queries or SMQs, particularly they're pointing out maternal and fetal disorders, which is a standardized Medra query that we have.
I'm not sure that I can answer that question. I don't know whether Brian, you might be able to answer that question.
Yes, I don't have a direct answer to that question. I don't know, Pat, if you're able to chime in, if you can add any response to that question.
Yeah, I think the SMQs have a slightly different purpose, where they're there to gather from a broad range of cases. So while I think there is some crossover between the lists, and maybe it's worthwhile to look at what's in that particular SMQ versus what is on this particular terminology, I think they have slightly different purposes.
Okay, great.
Okay, thank you. I will have a look.
Yes. Yeah.
Right. So next question is, will you extend on the many more fetal and maternal adverse event terms that are available in MedDRA to cover more, I guess, to cover more of what's in MedDRA?
So we weren't aiming to add anything which MedDRA already included. So the ones that we have produced were new terms. So the terms that were discussed with MedDRA back in 2016 were the terms that we developed the grading about. If we come up with anything that might be new terms, we will of course discuss it with MSSO and just see how they might map. Are they existing? Is there a way of are they actually new terms? I think there are potentially some new terms out there that we haven't considered. For example, fetal anemia, we can probably measure it using ultrasound Doppler of the speed of blood flow through the middle cerebral artery. But any new term in the fetus, it needs to be something that's diagnosable. We need to be able to detect an abnormality in the fetus. And I think as new techniques come online, we'll have more and more ability to do that.
Okay, thank you. So a couple more questions here. Next one is, should anemia of pregnancy be considered for the fetus also? If yes, then why not for other events like Eclampsia, which can have an effect on the fetus?
So yes, I mean, preeclampsia, well, the preeclampsia might cause the mother to fit, but we couldn't necessarily measure preeclampsia in the fetus. The preeclampsia might cause the mother to go into labor early, but it's probably not going to have an absolute effect on the fetus. But yeah, I mean, that's something that we probably need to have a look at. Are there other maternal adverse event terms that will affect the fetus directly?
Okay. So next question is, from the grading system that you presented, is it possible to have an adverse event that was deemed as serious with a grade 1 of mild? For example, is it plausible for a woman to have an early miscarriage that was complete, didn't require hospitalization or other interventions, but was deemed a serious medically significant event, and have a grade as 1 for the mother?
Well, I think it goes back to generally grading, if the mother is hospitalized, would be a grade 3. Now, of course, if she had a very light bleed, it could potentially be graded as mild. I think, yeah, I think it would depend on how much bleeding she had.
Okay, great. I think that's all for the questions. So again, we'll have a period after the rest of the presentations to answer any additional questions.
Well, I'm around for the rest of the time, so happy to answer them later if there are any more that come up. Okay.
Terrific. Well, then we'll move to our next topic, which is somewhat related on monitoring pregnancy outcomes. And we have two speakers, both from the European Medicines Agency on this topic. We have Cosimo Zaccaria, who works with the identification of adverse pregnancy outcomes in Eurovigilance, and Corinne DeVries, who is head of the Translational Sciences Scientific Evidence Generation Department in the Human Medicines Division. So Cosimo and Corinne, the floor is yours.
Okay, thank you very much, and good afternoon and good morning to all participants. As mentioned, this is going to be a joint presentation between myself and Corinne DeVries. Next slide, please. Before we start, we need to show the disclaimer for the European Medicines Agency. And with that clear, I think we can dive in the presentation. So next slide, please. So today's discussion builds up upon the foundation that EMA has laid over the past years to improve pregnancy surveillance. In particular, we'll talk you through some of the aspects concerning the management and the reporting of adverse pregnancy outcomes. And we'll focus a little bit on the retrieval strategy, which actually is creating a little bit of issue for better identify the pregnancy cohort within hypervigilance, which then could facilitate signal detection methodology. And I will hand over to Corinne, who will talk you through the some of the ongoing initiatives within the network. Next slide, please. So a little bit of background information. As usual, we mentioned the thalidomide, but really the thalidomide crisis created a pharmacovigilance system that became more systematic, organized, and regulated. So that now marketing authorization holders are requested to collect spontaneous reports and adverse events, which are submitted in the form of individual case safety reports, so-called ICSRs. So these reports are submitted and they cumulatively support the safety monitoring. And this is an important change that changed a little bit also the value of the spontaneous reporting in general, because a good pharmacovigilance system, from one side, should promote evidence generation, support authorization, particularly in the unmet medical needs, while ensuring robust monitoring for new safety issues, rapid decision, and effective actions, demonstrating a positive impact overall in the safety profile of a drug. Next slide, please. So we move now a little bit into the aspects of some of the aspects that are involved in the management and the reporting aspects of in hypervigilance for safety and but also for pregnancy outcomes. So in terms of management, spontaneous reporting is one primary source of information. This means that maternal and paternal exposure should be followed up, and information should be gathered on child developments over time. The coding of spontaneous reports should be compliant with the Medra versions, which is an important factor because it creates a structured database for querying for specific analysis. Additional data fields, however, can be used to routinely collect information. And in this particular field or area of pregnancy, route of administration, in this case transplacental, as well as gestational age, should be provided by the reporter in the context of the pregnancy case. These are recommendations, only a recommendation provided as a form of guidance, and they are built to overcome the absence of dedicated data field for pregnancy reports. And the lack of these data fields creates the challenge in retrieving the data. So we provided some guidance to overcome this issue. Next slide, please. So just a few words on what is reportable into hypervigilance in line with GBP6, which clearly defines what is either reportable or not reportable as an adverse or as an individual case safety report. So reportable ICSRs include congenital anomalies and developmental delays, fetal death and spontaneous reporting, as well as suspected ADRs in neonates. However, drug exposure in pregnancy without a suspected reactions can only be reported in certain specific situation that is stated in the RMP, for instance, for teratogenic drugs such as thalidomide and isotretinoin. In terms of non-reportable cases instead, these include special condition for instance, induced termination of pregnancy without malformation, pregnancy exposure without outcome data or with a normal outcome data. And these reports should instead be collected and discussed in the PSUR. Next slide, please. Yes, this is just to represent a little bit the decreasing trend over time in proportion of cases that are retrieved by the specific SMQ normal pregnancy. I just mentioned that such cases shouldn't be reported in hypervigilance. However, we do find them when we query our database. And so these events are basically not associated, as I mentioned, with an adverse event. And since 2017, we see a decreasing trend in line with the GVP6 revision, where specific guidance has been provided to collect such report in the PSUR instead. So hopefully the database is becoming cleaner in a way that we could focus on adverse pregnancy outcomes. Next slide, please. So let's move now to the strategy that could facilitate data retrieval and signal detection. So next one, please. In terms of the main objective of this study, we actually focus on improving or enhancing pregnancy surveillance. However, to be able to do so, we had to build an algorithm to be able to detect the correct course, the pregnancy course with Immunovigilance, at the same time assess or build some or explore or test new methodology to be able to confirm or not known association. And this is also part of the conception initiative. And but also we could use the algorithm to support descriptive analysis in Immunovigilance, looking at the most reported drugs and events in pregnancy, trends over time, and reporting over time in terms of quality of the reported cases. The algorithm is already implemented in the system, and we are monitoring the performance over time. In fact, we took advantage already from our findings, so our learnings, that we inform already the GPP pregnancy module. We also aim to create more detailed point-to-consider documents to educate the reporters, feedback on data input to industry and national competent authorities, aiming at improving data quality in general. Next slide, please. Let's focus now or dive in a little bit on the characteristic of the matrix of the strategies we have at our disposal to query Immunovigilance. Well, we have the gestational period data fields, which unfortunately is not giving us a large number of cases because this is only available when it's populated. We have also the parent-child reports, same story. A larger number of reports, however, not consistently populated or correctly sometimes. The miscoding issue is there for parent-child reports. And then, of course, we have the Medra terminology grouping that we could be using for grouping relevant cases. So we have two SOCs, system organ class, at our disposal, but also it seems that the SMQ it seems to be retrieving the majority of the cases. The SMQ is a little bit too broad, the pregnancy and neonatal topics. That's what we call level 1 SMQ. And it goes into other, not really, it's not fit for our purpose, let's say, because it contains different subgroups that they go also to lactation and breastfeeding, or as I mentioned, the normal pregnancy outcome. So next slide, please. We then come out with a case definition to be able to answer a specific research question. Is the SMQ good enough to retrieve all cases? So in terms of sensitivity, but also taking advantage of all the metrics available, are we retrieving the very same cases using different query strategy? And last but not least, how precise is this strategy we put in place? So we build up our own case definition, considering what we will constitute a false positive, or let's say a less relevant case for signal detection, and a true positive for signal detection, for which we believe that there would be an impact in the also in testing methodology. And this, of course, includes adverse pregnancy outcome that have an impact on the offspring, on the mother, or on both. Next slide, please. Here are some preliminary results. We went through a manual review of 100 random cases of the SMQ congenital and familial disorders to identify what are the correct metrics that are actually picking up true positive, but also what are the metrics that are creating miscoding issues in Immunovigilance, which are creating our false positive. As you can see from the green table, but also on the pie chart, you can see that the SMQ congenital and familial disorders, only 58% of the cases met our own case definition. and the rest were still, so the precision wasn't optimal. So we assessed also what was causing this false positive, and to understand what we could exclude from our algorithm to improve the performance overall. Next slide, please. So our next slide, please. Yeah. So from our perspective, actually, no, we should go one slide back. Apologies for that. It's a little bit delayed here. Okay. So looking at the available strategy, so our algorithm is a combination between specific major terms with a full inclusion criteria or partial inclusion criteria from the SMQ, and additional data fields, and that could be filtering on specific age groups, but also looking at additional data fields. But in this case, we removed 2 SMQ in terms of Medra terminology. We removed the lactation, as I mentioned, and the normal pregnancy condition outcomes. Next slide, please. So here is an example of the additional fields or data fields we included. The route of administration. We also excluded indication in case of contraception or lack of efficacy as contraceptives, and induced abortion, as these were not describing a specific drug exposure during pregnancy leading to a specific adverse outcome. Next slide, please. Yes, here it's in terms of descriptive analysis. So we run this algorithm through Pharmacovigilance to understand what kind of reports, and to compare also the reports retrieved by the algorithm versus the reports retrieved by the SMQ in light blue line, and then the report over time of the parent-child reports. Of interest, or actually of note, we can see that yes, the SMQ overall is a little bit more sensitive, is actually retrieving more cases. However, as it is a little bit too broad, that could lead to not a very precise retrieval. On the contrary, the parent-child report seems to be decreasing over time, and we are investigating the reason for that, whether some strengthening in the guidance or more precise information or recommendation could be provided there to support retrieval of pregnancy cases. Another important aspect is that the algorithm started to pick up, or actually to increase the trend over time, which is the dark blue line. As soon as the 2017 GVP revision was published, where non-serious reports are also submitted now to Pharmacovigilance, which could provide an important aspects of the general safety profile in pregnancy. Next slide, please. This is carry on the descriptive analysis. Other elements of consideration are indeed when we look at the trend over time of only cases picked up by the algorithm. The red line, as mentioned, this is increasing, which is reassuring, and is a good news because it enables us to perform a better signal detection. We also note in the bar charts, or in the blue and the light blue, we notice that there is a discrepancy between EA cases versus non-EA cases. But as from 2017 here, the distribution seems to be more even. And also, of course, as expected, the majority of reports concern post-marketing, as we know, many clinical trials in which we have safety monitoring in pregnancy. Next slide, please. Yes, this is my last slide with some data from Pharmacovigilance. This is just to flag that the algorithm has been employed during the COVID-19 safety monitoring crisis. Basically, we queried the database using the algorithm to monitor trends over time overall, and this could provide an indication of the increase in COVID-19 vaccination in pregnancy. In particular, we noticed a peak around this summer when CHMP recommended mRNA vaccines used in pregnancy. The algorithm was used also to provide some descriptive overviews, type of reports, consumer versus healthcare professional, serious versus non-serious report. However, it was reassuring to see that the most reported reactions seem to be in line with the background rates in this population. So no safety concern raised so far for COVID-19 vaccines in pregnancy. Well, let's say that the safety profile is very similar as compared to the others, to the other groups. Next slide, please. And this is moving to the end of my presentation. And as the next steps, we plan to finalize and publish the algorithm together with a description of the pregnancy cases in Vigilance. We would like to implement evidence-based methodology. This could be shared with the network, but it would be useful to see how this methodology performs in other databases, collect valuable feedback, and encourage reports to follow guidance and improve the quality of reports overall. And next slide. Yes, next, please. Here we go. And to conclude, spontaneous reporting is a primary and valuable source of information, readily available, has played a crucial role during COVID-19 safety monitoring, and it's proven to be very effective in identifying signals. The challenge in pregnancy is to correctly identify the right reports or the right cohort and develop new methodology, and EMA is moving in this direction. But I also like to flag that it's really crucial to have a strong international collaboration in this area, particularly in this area, so that we can establish an infrastructures that enables better access to safe and effective medicines in pregnancy. And with that, I hand over to Corinne. Thank you.
Thank you, Cosimo. Good afternoon, everyone. So I've been asked to complement Cosimo's presentation with a brief introduction to what is happening internationally in this space. And I'd like to start with the IMI project that's called Conception. It's a consortium. The abbreviation stands for Continuum of Evidence from Pregnancy Exposures, Reproductive Toxicology and Breastfeeding to Improve Outcomes Now. And the project is IMI funded, and that means it's a public-private partnership where there are 8 FPR companies involved. So those are the large European pharma companies with 36 public partners. So small to medium enterprises, but mostly government organizations and academic institutions. And they are building an ecosystem for better monitoring and communication of medicine safety in pregnancy and breastfeeding. And they want to do this in such a way that it results in a validated and regulatory endorsed workflows for fast and optimized evidence generation. So they really, from the outset, have been talking to the European Medicines Agency because they want to make sure that whatever they deliver is considered robust enough that it actually results in meaningful change that makes it into the labeling for patients. I don't have time, of course, in this afternoon to go into everything. So you have here an overview of the different work packages of what they're doing, and on their website you will see a number of you can go into great detail into what they're doing and what their events are. They do quite a few webinars to both disseminate their findings and collect from the public and other from all stakeholders input into what are the needs in Europe in this space. So I won't go into detail, but generally their work package one is moving beyond pregnancy registries, going more into electronic health records and any electronic recordings of health, societal factors that might impact on pregnancy outcomes and so on. Then the work package 2 is improving pharmacovigilance, both the analysis and interpretation, and that's where Cosimo's presentation of just now comes in, because they use pharmacovigilance systems and see what can we do better, both looking at the Medra terminology, but also other medical code books, other systems than eurovigilance, and seeing A, how do we harmonize better, and then B, how do we generate signals better, how do we interpret as well. So how do we analyze better, and how do we interpret better any signals that come out? Then they have a work package on better predictive models for transfer of medicines into human breast milk, because the current models are mostly rat-based and not that good, and they're developing different models that they're coming to the agency for qualification procedures to see whether whether they're on the right track, whether we agree with their PBPK modeling methods and so on. And finally, they're also creating a human breast milk biobank to support this information. I think for us as regulators, the key work that they're producing where they're entering into dialogue with us is, can we reach agreement on the adverse events of special interest? And so Anna David's presentation of just now, really links up nicely with that. And it's a shame that Anna David is not part of the consortium, but that is something where the consortium will be made aware of the work that Anna David has done, if it hasn't already been communicated. Then they have a core, so it's adverse events of special interest, core minimal data set required, where regulators agree that yes, if you have this data, then the data source, we agree, should be useful for monitoring the impact of medicine use in pregnancy and breastfeeding. And of course, it depends on the products and on the indication and so on. So that's going to be an interesting discussion. And then finally, on these non-clinical models for transfer into breast milk, we also have a dialogue with the consortium on that. So it's the project started in 2019 and it'll conclude in 2024. It's already being used in the COVID pandemic, for example, where we knew that people will be asked, these COVID vaccines will be developed. Can they be used in pregnancy, knowing that pregnant women are not included in the clinical trials? If treatments for COVID are being developed, then can they be given safely to pregnant women? And again, the clinical trials generally exclude pregnant women other than compassionate use, for example. So the Conception Consortium has been tapped into for looking at utilization of medicines in COVID in readiness for evaluation of any signals that might come up, and in readiness for evaluating doing simple association studies of pregnancy outcomes following exposures. And of course, even Europe is not big enough for that, especially now that we see that women are highly unlikely to receive treatments for COVID during pregnancy. So we're now also working in a global meta-analysis, trying to get a better handle on this. And of course, ultimately, the ambition is that we have better information generation throughout the product life cycle on the impact of benefit risk for the mother and the child in pregnancy. And so the big gap there is clinical trials. And there is increasingly a recognition that this needs to change without being cavalier. But at the moment, what we see happening is that a product is licensed for use in adults, and unless there is a specific contraindication in 4.3, of which there are not that many, then it's by implication licensed for use in pregnancy. And so we're saying basically we do not want to include pregnant women in clinical trials because of the ethical considerations, because of the sample size considerations. You simply won't ever have enough data in a clinical trial to demonstrate safety in pregnancy. But what is happening then is that the products are being used once authorized and not that systematically monitored. So you're creating a situation where you're almost experimenting without the due diligence of follow-up. And so what we're hoping is that both with the conception, that is going to change because there will be an infrastructure built to be able to monitor more consistently and more systematically. But also we're looking at, should we rethink how women are in or excluded in clinical trials? And that is going on at the global level, so in the context of ICMRA, WHO, and ICH. And I think, you know, so in that we're then bringing together the learnings from conception, from the COVID pandemic recently, from treatments with HIV, from the fact that increasingly people with chronic diseases are living longer and are becoming pregnant while on treatment. So yeah, there are lots of opportunities to improve. And I think that's all from me for the time being, and happy to take any questions. Thank you.
Okay, there are no questions at this time. As I mentioned, we will be having an open question session at the end of the last presentation. So there are plenty of opportunities to ask questions on this topic. So we'll now move to our next presentation, which is on the topic of pharmacovigilance experience and challenges of COVID-19 vaccines in Germany. And that will be presented by Dr. Dirk Menzer, who is a consultant in general pediatrics and head of the pharmacovigilance unit at the Paul Ehrlich Institute. That's the Federal Institute of Vaccines and Biomedicines in Germany. So Dr. Menzer, the floor is yours.
Thank you very much, Brian, for the brief introduction. And I will just give a few backgrounds on myself. I'm a pediatrician, and I will be now working 17 years for the Paul Ehrlich Institute as the head of pharmacovigilance. In the meantime, I had a number of years working as a delegate at the European Medicines Agency, and I've been always involved in pediatrics with that, based on my training. And I was 6 years chair of the pediatric committee until 2019. And now you can imagine over the last 2 years, I was mainly involved in pharmacovigilance of COVID vaccines. And this is the summary a little bit leading to my topic, where I would like to show you some of the preparation for Germany, before starting the campaign, and of course sort of listing the challenges we had and the experience for that. So this will be sort of basically the flow for my talk. Just a brief disclaimer: I'm a member of a federal agency in Europe and I have nothing to disclose, and of course all this is my own property from thoughts and everything presented. When we started preparing the COVID-19 vaccine campaign, of course, we didn't actually really know when it will start in the beginning. Eventually, it was as early as we thought. And we knew from the beginning that this would be a challenge because we were facing new vaccines, the vector vaccines and the mRNA vaccines. So we also knew that we will not have a lot of long-term data due to the short development phase, of course. And we also knew that due to the potential prioritization, we will start in a population where we have little experience, neither from the studies or from any other prior vaccination. So here you can easily understand how challenging it was to prepare something, as we were sort of going into the darkness with all the knowledge we had. And of course, what we were also realizing that mass vaccination will always cause a huge amount of reports. We were trying to match this to the 2009 swine flu vaccination campaign at that time, and we knew it was a lot more than we normally had, but this, of course, something we had to count in all our preparations. And finally, the only way we thought could go on, that we need to have some study designs. We have to need some implementation of studies to overcome also the unknowns in this starting vaccination campaign. Besides that, we had, of course, preparation with the routine pharmacovigilance here, a strong collaboration with the European Medicines Agency, and in the end, the list of adverse events of special interest, I would say, was a huge investigation ease for us in the end to look into the adverse events on a sort of really magnifying glasses and have a sort of predefined set of data to look at. Also, the numbers of case definitions from Brighton population, which were updated or renewed. If you think, for example, at the TTS symptoms, we then sometime for a year ago had to face so the thrombocytopenic thrombotic syndrome. This was also very helpful, of course, in this collaboration. So here, I think the regulators all had a really good approach. Once we saw the huge amount of data coming in, we knew that PER was not able anymore. The signal detection using the first as expected analyzers become the more standard now than had been before, but all the ups and downs looking into the background data and all this. So this, of course, was a huge change in our routine pharmacovigilance we had. And then before I start a little bit into the way we have to overcome all the challenges, I need, and this is also linked to the Medra coding and the matching we are talking later in my presentation, is the way we have a reporting system in Germany. So we have basically the usual reporting regulation obligation from the marketing authorization holders to Eurovigilance, of course, and we get the backlog data, the rerouting data from Eurovigilance into our database. So in the end, we will see it. And this is since 2017 that this way goes through via Eurovigilance back to us. But we also have the healthcare professionals and, of course, increasingly during the pandemic, really increasingly, the consumer or the affected persons following vaccination. And the healthcare professionals, for example, they have a legal obligation in Germany to report through our local health authorities, and they have to report to the pharmacovigilance system in our agency. So here you see this is something we really thought would be quite helpful for us to have this kind of quality data from the healthcare professionals in that way. In the end, I have to say that 90% of all our reports came from the people being vaccinated directly. So, of course, it has ups and downs, and you can imagine they're not reported in any Icd-10 or Medra coding or whatever system. So we had to do a lot of recoding work in that direction. So what did we do for our to overcome all the challenges? We had basically designed one cohort study. This is based on a smartphone app, and I'll show you that later. And then we had, of course, a number of retrospective study analyses, and one has been started now in just end of last year, basically, because we were hopefully starting it earlier, but due to the data protection laws in Germany and the obstacles we had to actually get the data from the insurance companies were quite dramatic. And I have to say that this is taking a long time to overcome all the problems. As parallel, we have started the pregnancy registry with the Pharmacovigilance Center in Berlin, the embryo tox center of the hospital Charité in Berlin. And this is running quite well, and they also not only do the registry for pregnancy and then sending it via E2V to our agency, but also they do consulting of questions from pregnant mothers. So this is basically the special pharmacovigilance we have implemented. And besides, of course, that, back to the routine pharmacovigilance, we had to update our online reporting platform which is from our website reporting to our internal E2B database. And here we have implemented a specific COVID-19 reporting platform. So not really to sort of streamline or connect the special data related to the COVID-19 vaccine, but basically to sort of get some hands on the different typings. And you cannot imagine what all typings we have seen for vaccines in that time. So that was quite a challenge to actually know which vaccine in the end was used. But what I'd like to show you is that we have used this platform and implemented a sort of matching aspect in that, specifically to our experience, which was then eventually also updated during the time on the vaccination reports. Also, adverse events of special interest include not here, but the IEFIS should have here, which were tried to match to Medra. So once the person who actually entered one of those codes in the background, the database has matched it to a Medra code, which then easily could be transformed to our E2B database. So in the end, at the highest, we had about 1,600 reports per day, of which we have to only about 10% to be recoded. So this, otherwise we would not have managed the whole challenge of reporting here in that system. The other basically reporting system we used to look into also signals was our smartphone app. And we had a study to collect all the adverse events following vaccination. And as this was a study design, so you had to log on once you were vaccinated and then report according to this time schedule here seen in brief, what happened to you post-vaccination. And I can tell this 75% of all participants had at least one reaction, which was surprising, but of course, most of the reactions were, and I'll show you the numbers later, what we've expected. So basically what we knew from studies anyway. So here we had quite a good representation on the actual real world evidence of vaccines being used through this app. Later, once the vaccination was approved also for the adolescents, we opened the study also from the 12 years and older. Of course, in the beginning, it was only from 18 years of older and older. All the reports were voluntary, so they had to be done in electronic consent that all data are sent voluntarily to our agency. We had altogether a follow-up of 28 days post-vaccination dose, and we are still collecting data on 6 and 12 months post-vaccination. So the study is, for some of them, still ongoing. We had introduced a monitoring for the booster dose as well, which was initially not part of the design of the app, so this was a little bit of a problem. But we managed then in November to also include this recording. The sample size is not limited, and I can tell you that we have 750,000 participants at the moment. We cannot identify the participants. This is a little bit of downside what we realized, but still this was an obstacle we had to give out for the data protection law we have in Germany. And we can, of course, manage and analyze the results. We have done that, and for example, in the 750,000 records, we have about 60 records for myocarditis. So, and when you break down to the age decades, it is not new what we see, but it reflects what we know from the Australian data. So those are the data I like to show you. Interestingly here, based on the readout on 29th of April, female and male participants are almost equal, whereas when we look into our data by spontaneous reporting, 2/3 of the data are from female and 1/3 are approximately from male. So for some reason, this is an interesting difference here. The other interesting thing is that when the seriousness of the reports are by self-reporting, and it is 0.3% in the whole population we have been looking at. When we sort of match them with the adverse events of special interest, which per se have to be reported as serious, it's much higher. So it's about 3-fold higher, but still approximately 1% would be counted as serious in this means. Then this is the age distribution, and not surprisingly, we have quite a huge number over 70 and 80. You wonder sometimes, the oldest is 104 years old lady. Of course, we think that not herself is reporting, but still the participation from the old age is also quite well represented here. And this is a brief update on the adolescents reports we have at the moment. Is only started in November last year, so we didn't catch enough, because at that time the majority of reports, the majority of vaccination has been done, and as we only had 2 days post-vaccination to enter the study, as we wanted to keep it as a prospective observational study, a lot of adolescents could not enter once they had been vaccinated, according to the availability of vaccination. And this is the interesting thing we also monitor, of course. But if you remember the headings I had in the listing from our spontaneous reporting system, the same were entered as a pre-selected reports in the app. So of course, those are the majority we know from if you look in the SNPC, and if you look then into the lower parts here, it takes a long time till you see some of the really severe events. We don't have any TTS cases, for example, but this is not surprisingly, but because we don't expect to have the hospitalized cases in a representative manner, as people will probably not report once they're in hospital. So this comes to the end. So in the end, what we have experienced in our preparation, that using matching systems, for example, I showed you with our spontaneous reporting in the app, in such a vaccine campaign population-based is more or less mandatory if you want to overcome the huge amount of reports, and it helped. And the next step we are going to do will be the consumer reporting platform we have. We will have a split platform for healthcare professionals, and potentially there we are expecting more ICD-10, because that's what they use for their coding when they go through the diagnosis and reporting to their health insurance. So this will be the second level of coding matching we will use, apart from the direct coding matches we have for MS for Medra in that area. The second thing we had done for transparency, we have always in the beginning weekly and then monthly and now quarterly have reports on the outcome or on the reportings what we see from the vaccine nation campaign. And in this protocol we published on our website, we always had also reports, for example, on our Sapac outcome to give a monitor on how things are ongoing with that app study. The final analysis of the app study is awaited now, because we will need to wait a year following the mass vaccination. Will be probably in August, September, when the vaccine campaign eased. So this is the plan for that. And the other plan we will sort of try is to increase the availability of the app, not only for the study, but also for the spontaneous reporting system here. So that's me for our overview.
Okay. Well, thank you, Dr. Metzer. We do have a couple of questions. The first is, how did you confirm, verify the reported adverse reactions, especially the more serious ones that were reported by the public through the Sapac app?
Yes, as we cannot track the reporter, of course, the validation here was not on person. So we had to see whether other possibilities are matching to the report. So that was our basically our ability to vaccination validation of the reports, yes. Spontaneous report from the website, of course, we can track them if they have mentioned their contact addresses, and we will, of course, try to record the validation by either medical records they can send us or try to contact the physicians.
Okay, thank you. Next question is, what were the adverse events of special interest monitored or detected by the Sapac app by self-reporting?
Well, basically the adverse events of special interest, only a few were in our list, in the predefined list. Basically, the people were free texted, so they were able to, for example, myocarditis was not in, so they had either the German version for myocarditis or they even used myocarditis themselves as a word, and this was recoded, so all the data from the app went electronically to our internal database, the E2V database, and the E2V database detected free verbatims on reports. And on that part, we have sort of an artificial intelligence module in our database, and it suggested to the colleagues who are doing the coding in our unit, suggested already Medra codings on those free texts. So we then would be recoding the free text into any Medra code, which would be suitable then.
Okay, well, thank you. So there's no additional questions at this time. Again, we will have a Q&A session at the end of our agenda today. So feel free to ask additional questions.
Yes, I will be here.
I'm sorry. Our next speaker is Ian Slack, who's the Director of Clinical Data Management at Vertex, and he will be providing a short presentation on a recent medical coding survey that was conducted and the plans to provide and discuss survey details. Ian?
Thank you very much, Brian. Can you hear me okay?
We can, thank you.
Excellent, okay. Well, good afternoon, good evening, good morning, everyone around the world. This presentation is a really short one, just to whet your appetite on what's coming next to you regarding a medical coding industry benchmarking survey that was done last year. So if you can move to the next slide, please. So basically, we were looking at what was out there in the industry. There's lots of metrics and data for various parts of our clinical trial processes and the industry in general, but there was very little out there for medical coding. And so we took it upon ourselves. We had a collaboration between the EU Industry Medra User Group Steering Committee, UMC and the MSSO to develop a survey of the industry. It was very evident initially that the scope was quite big, and so we had to narrow that scope down, and we looked at the metrics for coding standards and processes and technology usage in the end. And we uncovered quite a lot of excellent details that we will be presenting to you. So the survey was carried out between November 2021 and January 2022. And representatives from 20 unique organizations took part in that. It might seem like a small number, 28, but actually that represents quite a number of coding personnel around the globe. And we didn't want to have some duplication of input, and so we were very cautious around how we were going to collect that data. The results will then allow us to look at benchmarking where we currently are in industry with things like auto-encoding hit rates, error rates, use of certain technology, and other ways of managing quality and our processes in the jobs that we do every day. And we wanted to open this up to not just look at clinical coding, but also pharmacovigilance as well, and open it up to all possible usage in the industry. Next slide, please. So how are we going to present these results? Well, we wanted to, rather than giving you spreadsheets and Excel documents with all of the data in there, we wanted to crunch the numbers for you and present the data in a subset. Some of the information that we received didn't really provide us with a lot of what we call significant feedback. And so we took the main outputs and the key takeaways to provide these quality baseline metrics for you and to summarize the feedback. It wasn't just about numbers, it was also about interpretation and how are we going to potentially improve where we are right now, what we're looking for of the future. So we thought we'll do a presentation of the information, but also have a panel of experts to discuss some of the findings and to interpret that and really sort of look at how we can use the data that we've received and what we can do next, what we can use that data to do. So that panel of experts consists of Martin Menk from CSL Behring, Hank Dennis from CGen, Henry Chen from Thesis, and Mansi Joshi from IQVIA. I'll be moderating the session, and we're going to be presenting this at the Who Drug and Medra Global Webcast on the 22nd of June, and you will be receiving invites very soon. So I hope now your appetite has been whetted, and you look forward to seeing what this data reveals and what we can actually do with it. And I'm open to any questions.
Okay, thank you, Ian. Does not seem to be any questions at this time. So I think what we could do is, oh, hold on, I take that back, we just got something. Will the survey be published as well as presented?
The slide deck will be published with the information on it and obviously there will be certain metrics that we present there. We're not going to be presenting or distributing all of the input that people made like in the form of Excel or anything like that, because we didn't want any sort of identifiers, or we wanted to make it completely anonymous and summarize the data per the information that we or the proviso that we gave out to the contributors.
Okay, thank you, Ian. Right, I think that is all the questions on your topic. What we can now do is move to the last item on our agenda, which is the open question session. So this is the opportunity, if you had a question on any of the earlier presentations, or you have a question in general for the MSSO, this is your opportunity to ask that question. So we'll wait a short period here to see if any questions come in. We do have one question that just came in. I believe this is for Dr. Menser. I'm just going to read the question as stated here. I'm not sure what's meant by part of the question, but we'll see if we can handle it as best we can. The question is, have there been a lot of politically motivated false serious AR adverse event reports, for example, myocarditis following COVID-19 vaccination?
Well, that's an interesting question, of course, and this was this is an ongoing issue. Of course, we have been asked from our political parties as well during the last weeks and months. I just can say that from the reporting pattern, and here it's not only the amount of reports we have to a certain population, which would you expect basically that somebody tries to jeopardize the reporting system by sending huge amount of reports of into 40 population, for example, or even the adolescent population. I cannot say that we see that, and this may be due that when you think of spontaneous reporting, not only via the app, of course, and it may be easier via the app than others, but if you think, for example, from our website platform, technically the platform is secured in the way that you can only log on a certain times in a certain time period, for example, so that would the Robota reporting, for example, would not be able to do so. And then, of course, it's when we see reports, and we do see a few fake reports, which we think are fake reports. For example, a 2-year-old being vaccinated, having 78 centimeters, 107 centimeters, 18 kilograms, something like this. This, of course, is invalid. Yeah, so this is not a 2 year. So somebody did write something just by chance. So we do see those reports, and of course they're selectively really one-off cases. So we can say we have been hacked once. One reporter tried to hack our smartphone app. It was defended. So the ability we used to defend this was as much as we could, of course.
Okay, thank you. All right, so at this point, there are no additional questions. I'll just give it just another few moments. Oh, here we go. Okay, several came in. Let's see here. Okay, so this may be either for Professor David or our EMA colleagues. In the clinical trial setting, should pregnancy collection and pregnancy follow-up be done for study participants that have not received the study drug? There is not any study drug exposure. So I believe that would be for our EMA colleagues. But Dr. or Professor David, you certainly can chime in on that as well.
Sorry. So thank you for the question. I'm not sure I understand the question. So is the question, if someone falls pregnant during a clinical trial and they were not on the study drug, then should they still be followed up for the, should they still receive additional follow-up for pregnancy, adverse pregnancy outcomes? Is that the question?
I believe so. I just, it was just entered in the chat. So you can take a look at the question.
Oh, yes, sorry, I haven't been looking at the chat. Okay, in the clinical, should pregnancy collection, pregnancy follow-up be done for study but not received? Yeah, yes. So yes. I think, to me, there are a number of scenarios. One is if you have a clinical trial where the indication for prescribing is specific to pregnancy, then there is the scenario where you have people who fall pregnant during a clinical trial and it's unplanned. And third, what was the third one? I've lost my train of thought. And basically, what we're getting increasingly is questions where women want to be enrolled in a clinical trial for a product even though they're pregnant, or they fall pregnant and they don't want to come off the study drug, or they fall pregnant and the drug has such a long half-life that you that the exposure in pregnancy has happened and will continue for some time. And so to me, then the thing to do, irrespective of whether the person was on placebo or on the study drug, in all of these scenarios, follow them up completely, give as much, collect as much pregnancy outcome data as possible. And in that sense, I found Anna David's presentation so helpful. Okay, what do you actually monitor? What do you find? And I would do that both for exposed and unexposed, because the numbers will be so low that you have, we won't find, it's highly unlikely you find something that's statistically significantly meaningful. You will find, because of the additional monitoring, you are likely to find potentially not necessarily even adverse, but deviating outcomes just because you're monitoring super carefully. And by doing that both in the exposed and the unexposed, what you would hope is that it balances out between the groups. And so to just do it for the exposed, I think that would be a mistake, because you might be causing undue concern about the products when it's not really when that's undue, basically. So I think collect as much information as possible, offer the women the support as much as possible, because they will be scared. What is the impact on my baby? And offer to monitor very carefully the baby and give extra support, and actually use the opportunity to learn about the psychological impact, how best to support, how best to communicate uncertainty in these situations, because it's no longer uncertainty about benefit risk for just one person. It's a person who feels extra responsible for their child who may have benefits and risk associated. And so I'll stop there, because I'm sure Anna will want to add.
Yeah, Corinne, I think you're absolutely right. I think it's very important, first of all, that we do keep continuing to collect up information from women who've taken part in a trial when they get pregnant. If you were to not collect up the information, it may potentially send out a message to the people who are continuing on the active ingredient that you're more worried about them than you are about the women who are in the placebo, and that may be absolutely untrue. And I think that was some of the messaging that came across from vaccines was not, we don't know what the risk is, but you shouldn't have it because we haven't tested it. And so I think our patient group were very clear. They felt that it was important to collect up all outcomes, regardless of whether you're on an active ingredient or on a placebo. And obviously, if you get pregnant during a clinical trial, if possible, if the clinical trial protocol allows, to continue to collect up that information. But I think we have to be very careful about messaging correctly to patients so they don't get the wrong idea. So I totally agree with you, Corinne. Continue, if you can, to collect it up.
Okay. Let me pivot to another question that was just posed, which may also be in the same general arena here. The question is, in the clinical trial setting, we often face challenges for certain adverse events on neonates where congenital abnormalities are serious criteria, but the condition is not considered serious by doctors. What is your comment on this? I'll put this question in the chat.
I can try to take this. Well, in terms of reported rules, as I mentioned during my presentation, non-serious reported 2017, now part of the GBP6 revision, should be reported to the revisions because they allow, defining a better safety profile, and that is a recommendation. So this is part of the revision. So that is reportable as long as there is an event associated with the administration of a drug, then it should be reported.
Okay.
I'm just wondering whether that was the question. So for certain adverse events on the neonates, congenital anomalies are serious criteria, but the condition is not considered as serious. Oh, by doctors, is that what you mean? So that there is a congenital malformation, but it's like a 6-digit or something, and that's not considered serious. Is that the question?
You can ask the submitter to confirm if that's the answer to the, or that's the frame of the question.
I think that's probably why it's so important that we collect up information from people who are not exposed to drugs, because, you know, these kind of potentially non-serious congenital abnormalities are relatively common. And so it's why it's so important to have non-interventional control data or placebo data, so that we can, you know, really get a handle on how common, and congenital abnormalities are very common. And in some populations, they're much more common than others. You know, where there's consanguinity, you may help to about sort of 5 or 6%. And so, yeah, it's important to collect it up.
All right, we have another question. I'm going to send this in the chat as well. The question is, some sponsors want to collect pregnancy data from the day of informed consent, and even from the prior first dose. We see pregnancies with clinical trials prior to their first administration on the study treatment. Should those pregnancies be monitored until the outcome as well? As well, yes. I put it in the chat. So any comments on that question?
So this is the intention to treat question. And I'm very much of a get as much information as possible. You will have people with a disease of interest who are receiving treatments for this disease, and we know so little about the disease and the impact of the disease when compared with treatment. So I think diabetes is an example where we are increasingly comfortable that actually, as long as disease is managed very carefully, then the adverse pregnancy outcomes likelihood is lower. So that any adverse events tend to be because of poorly controlled disease rather than the medicines treated for that disease. We don't have that information for a large majority of disorders. So I think that is one reason. The other is it depends on what products this question would relate to, because so at the moment most trials will routinely exclude a woman like this, where there are situations where you could imagine, well, the standard of care is actually only consists of products that are teratogenic. So should you look at the non-clinical data for the investigational treatment and consider whether that gives some reassurance that possibly it should be considered to give the woman the treatment, or the investigational new treatment, rather than the standard of care, or in addition to the standard of care if the disease itself is associated with poor pregnancy outcomes. And I've come across a number of examples there. And perhaps we should have that dialogue more with women, their healthcare professionals, rather than just a routine exclusion. So my answer would be yes, monitor everyone and actually start to engage more in those dialogues. Thank you.
Great. So the next question I have is for Dr. Menser. This is regarding the use of the mobile application. And the question is, how well has that mobile application been accepted by health professionals?
Increasingly, we have, as I showed you, what our reporting system, the healthcare professionals have to report to the local health authorities. And of course, some of you may know that in the federal state like Germany, a federal agency also have to stand back behind the local agency, as is according to the federal system. What we are at the moment, we of course encourage, and we have the ability in our web system that the health can.
Your voice is breaking
Professionals
up a bit.
Can, of course, also, and those healthcare professionals, they also send reports to the local health authorities. So maybe the. Okay, I stopped the webcam. Maybe it's
yes.
better now.
Yes, thank you. I think let's try that again if you could.
No, what was time? Yeah. Okay. Yeah. It's a yeah. Okay. You know how I look like. Okay. So what I'm trying to say is that our web system has the ability for the physicians to report, also identify themselves as a physician report, in the part where you give your identification. So from that point of view, they can report. They can use this report. And also can report through that to the local health authorities via paper. Unfortunately, not other than that at the moment. But our experience is that, and that's what we hear also from anecdotally from patients if we talk to them, that the physicians say, yes, you can report. There's a website from the agency, and you can do the reporting. Yeah. So we have a huge number of those anecdotes. And of course, when we have the consumer or the affected person reporting, we then, in certain cases, of course, can ask through them back to the physician and then get additional information like hospital letters or so. Yes, but I think the workload in our healthcare professionals, they try to move it over to the patient or the vaccinee. But it's possible. Yes.
Okay, thank you. Okay, so let's see. Next question, I think, falls into, I guess, regarding the, I guess, question about pregnant women. Will you have long-term reports after vaccinations? Well, what will be the next actions for pregnant women? So I'm not sure if that's completely clear. So if it's not, I'll ask the submitter to restate that question and perhaps we can circle back to it. Okay. All right, so one other question, and I think we may be able to, oh, I'm sorry, Corinne, did you want to try to answer that?
Well, I'm just thinking, trying to interpret the question. So you still will have long-term reports after vaccination. What will be the next action for pregnant women? I'm interpreting that as, okay, so at the moment, there is no information from the clinical trials. There is little information from post-authorization, although I think the CDC in the United States has done a lot of work that's already out on their website. Of course, we have asked the companies to do post-authorization safety studies, and there are also some efficacy studies. And of course, we've got our consign work. So those study results will be coming in. And based on those study results, I imagine that we will be asking companies to update the label, because if the study results all say these vaccines are safe, and we know, because we're seeing many studies of the impact of COVID in pregnancy on the mother and on the child, then I would imagine that the labeling will be less cautious and more encouraging to women, that these vaccines are also, the benefits outweigh the risks also in pregnancy. Of course, if it's the other way around, if we get a surprise and we see unexpected adverse outcomes, then the labeling, will also reflect that in a different direction. But, so we are monitoring this, and we will be, when we get this, we will assess the study reports and have recommendations for the companies as to the impacts for labeling changes. I hope that answers the question.
Okay, thank you. One last question, I think then we'll wrap things up for the day. This is just a follow-up to one of the questions that was asked earlier. My question is, how do you face the challenges from investigator for seriousness evaluation of some congenital abnormality events? There may not be a perfect answer to this one, is my, I suspect.
I think this one is for Cosimo. He's our signal detection expert.
So again, I'm not sure it's very clear to me the question. So how do we assess the seriousness of such anomalies? Well, this is what we do with them. So we need to rely on what has been reported if we are referring to the case reports we assess. So we need to rely on what the reporter recognized, and for that we have case definitions. We rely on the case definition, and if a particular case meets the case definition, then this will support basically the next step in our evaluation. But in terms of signaling, and I'm not sure that it refers to the signal detection strategies, we also, of course, prioritize serious report, major information leading to fatal outcome and so on for review. But we mentioned many times we need to have a background to be able to, we need to have a denominator to be able to identify specific signals. So the seriousness is just one of the criteria we apply for prioritization. It's not the only criteria. So I'm not sure if that addresses your question.
Brian, can I say something to that? Maybe from the point of view of primary field collecting data. I mean, seriousness is really, really difficult to define seriousness from the reports. For example, if you get patient reports or personal reports, they say, "Oh, it was serious." Yeah, but if you then look back, they have never been in hospital. It's not so. According to the criteria of seriousness we use as a regulator, it's different from what the patient uses or the affected person uses or whatever. So I think here, and we've seen that through the report of the COVID-19 vaccine campaign. And there, as I mentioned, for example, in our app, the self-reporting seriousness is very, very low compared to what we normally see. But if you look then into the adverse events of special interest, which per se were defined as potential serious, and I know we have other important medical event criteria, which also included the congenital disease and at the EMA Medra listing we do. Those are also potential serious as medically confirmed, basically. So I think here talking about seriousness is difficult. It's important for us, of course, when we start trying to record additional events, additional information on those, yes. But Cosimo, please confirm, others I'm wrong. But for example, for signal detection, specifically in vaccines, the seriousness criteria is in the background, basically. So, for example, if you have a huge number of events, non-serious, this could be also difficult for a vaccine, for example, in a short time period. So I think here seriousness criteria is not really what we primarily look, because we have to look at them when we start the reporting timelines and all this, yeah, so get them on as quick as possible for the potential seriousness, and the non-serious, they can go later. But for example, if you have a non-serious case report, and then eventually, for example, tachycardia, and then it comes out the tachycardia is related to myocarditis, it changes suddenly from non-serious to serious. So I think here, going back to the serious criteria from the reporting system, it's difficult. It has implications on reporting timelines, yes, of course, and for us, implications on retrieving additional information, yes, but I would say for signal detection in the primary analysis, we look at all cases.
All right, and Professor David, did you want to comment on that as well?
I was just going to come in and say that the neonatal adverse event severity scale has a generic scale which allows you to grade things, but not specifically congenital abnormalities. But it has a grading system which goes from mild all the way up to life-threatening. So that is potentially a way to do it, but that not specifically for congenital abnormalities, but it's a generic one.
All right, well, thank you very much. So we are at the top of the hour, and this does conclude our meeting today. First of all, I'd like to thank all of our guest speakers for these wonderful presentations. We really appreciate your contributions in making this a successful meeting. A number of you have asked about the presentations for this meeting, and I have to apologize. My intent was to be able to provide those presentations before this meeting, but unfortunately we did have a few last-minute modifications, and for that reason I held off. We will be providing these presentations on our website. In addition, we'll be sending a post-meeting email to you to take a survey, and within that email I hope to include the link to the presentations. So we will have them available to you shortly after this meeting, roughly 24 to 48 hours afterwards. So with that, I thank you very much, and I look forward to seeing you at the next MedDRA user group meeting.